Hi, my name is Ben. Thank you for giving me the chance to present our work of decision prioritization using network and feature here. So this is actually a collaborated work with University of Chicago and also with the Toyota Technology Institute of Chicago. So the decision prioritization, it basically is a founded area. So basically we want to identify the disease genes based on we want to rank the disease genes, and so that we could feed the most confident gene into the wet lab for testing. And it's challenging because usually we have hundreds of, thousands or thousands of candidate genes. And we want to sort those candidate genes by the similarity with some no association, like no disease genes or no phenotypes. And the knowledge we have here is actually we have annotation on the gene or their products or like the interaction between the genes. So final goal is actually to narrow down the candidate gene to like a handful of genes. So that's like the ultimate goal for us. So it will be easier for the wet lab to test to save the cost and also the time for it. And so there's a lot of existing approaches here. So we break it into two categories. So one is the feature-based prioritization. So basically using the annotation features to do the prioritization, such as the top gene Denver suspects. And another category is actually the network-based prioritizations. And they're basically using network to get those similarity based on networks. And so there's a lot more tools that are presented in the gene prioritization portal, so feel free to just check that out. So there's still some problem need to be addressed. So as we know, the genes features are very sparse. So that means some of the genes may not be well annotated. And so in this case, there could be some of the genes which are novel genes, but they're not yet been well annotated. So that in this case, some of the tools might fail because there's no certain features, for example, the feature-based prioritization, they don't have annotations. So in this case, the approach will fail. And then sometimes they transfer the information between the features and networks. So they have those functional association network or the network being clustered and then annotated as features. But in this process, there's the data loss will happen. So we want to build a model which could modify both information at the same time. And also we want to build a specific model with respect to the user input. And as we know, the current, all these tools, they have decent accuracy, but still, there's still a lot of false positive on the top. So we want to minimize that to give a more accurate predictions on the top of the rankings. So our approach is actually integrate both feature and network in their own original form. And we are allowed the model build specifically to the user input, and also we want to reduce the false positive on the top results. And here are the normal features we have. So we have the functional categories like gene ontology, molecular interactions, and also the disease information, context information, and many more. So this is just overview for the features we're available here. And so we're going to give an overview for our workflow. So the first step is actually we took a list of user input features, genes, and then we extract the most critical features back to this group of user input. And then we combine the feature network and do inference using the conditional random field, and then we integrate from different sources and have the combined score. And the final step is estimate how significant is the score with respect to a general distribution of scores. So we'll talk these precision details. So the first one will be annotation and enrichment. So basically here, we're just extracting the importance of certain feature, for example, the goal terms, with respect to a subset of genes, which are from user input. So here we're using the links, which is our online tool. And we use that as the first step to do the enrichment. So it's available and links.seattle.chicago.edu, so feel free to explore it. And the second step is we're modeling the gene and disease association based on the features we're just getting from the enrichment and also online network. So let's suppose we have a smaller network with 6 genes, 7 edges here. So the usual CRF model will be parameterized based on the cliff we have. So here we are just model two kinds of clicks. So one is the single node and the other is pairwise nodes. And then we call these factors. And so the factors could be on the genes and also on the edges. So the factor on the genes can be furtherly parameterized by the features. So suppose we have the feature one as some gene ontology terms here. So basically it's further being parameterized by a group of features to the factors. And for the edges, we model it using like uniform probabilities to modeling that the two nodes in sharing one edge, do they agree with each other or they do not agree with each other? So we give a favor to, so that we say the genes tend to agree with each other so that they tend to have the same labeling, like they're both associated with the disease or they're not associated with disease simultaneously. And so we give a favor to that. So we're modeling that using the summation of all the features here, and we weighted that. And the weight we're using is extracting from the enrichment, so it reflects the importance of the certain features here. And then we'll do a global inference to get the probability of like the testing genes, like how likely it is related to the training genes. So it's the how it related to the diseases we're interested here. And then the third step is we're doing a data integration. So basically we're doing a linear combination of the probabilities from the label of each gene so that we have a combined scores finally. And then the next step is actually doing an estimation of how significant this combined score is. So we're using a large scale simulation of 10,000 times randomization of input genes, and then we obtain a distribution, and then we find the p-values for each score, and then we do FDR corrections on top of that. And so before I introduce the results, I want to first talk about the benchmark we're using. So the benchmark is actually from a paper with unbiased evaluation of gene prioritization tools from Daniela Borningen. And this is an evaluation paper consisting of a benchmark and with 42 target genes, and so one for each diseases. And the target genes have the characteristic which they're discovered after the certain time. And the training genes are actually the disease gene discovered before that time. And the testing gene we have is target genes, and we mix that with some the neighborhoods in the chromosome. So here we want to mimic a natural way of discovering the novel genes, which we don't know like previous informations for those target genes, and we want to rediscover it, and we want to actually see those target genes ranking on the top of the list here. And so that we're using the data-backed data sources to prevent that we have some prior knowledge on these target genes. So, the string network we're using is actually before 2010. Also, the gene ontology terms are dated back before that certain time period. And so that we compare our method with these two different tools. It's actually mentioned in that evaluation paper. So actually, these are the two best performed tools actually across a wide collection of tools. And one is pure feature-based and one is pure network-based, and they're using different data sources. So we show that so here is the Roc Curve of the three tools. So the blue line is for our results, and the red one is for Endeavor, and the green line is for Pinta. So we show the Roc Curve, but we zooming on the top 20% of this Roc Curve. So as we know, the most critical part lies in the top ranked genes. So that's we're most interested in. So we're zooming actually on the top 20% and show that we actually have a much significant higher error curve than two of the other tools. And so let's look at the in detail. So we have more like true positives. At the top 10%, we're much more than the other two tools, and also at top 15 and top 20. And the partial error curve is actually much significant than the other tools. So someone may say like the curves, they have higher accuracy, they might be related. So it's kind of trivial, maybe it's just like combine the features. So want to prove that we're actually complementary to the tools we're seeing here. So we compute the pairwise correlation with respect to the ranking of the target genes. So actually we're not correlated with both of the tools. And from the distribution of the rankings, we could actually see that we're much stable. And so the variance is smaller than the other two tools. And the worst case is actually like it's ranked as 50% ratio. It's actually much better than the two of the tools. So that means our approach is not like trivial with respect to the previous tools here. And we further look into the absolute ranking for the target genes. So seeing that we are able to identify, to rank the target genes at top 1 in, I think, 8 out of 42 cases, which is like 25% of the cases we were able to extract that on the first place, the target genes. And so in this case, we also proved that we could somehow bring out the most hidden information actually burying in the information we have with respect to the disease associations. And then we did some actual analysis with data sources. So currently, because we have a lot of in-house data sources here, like gene ontology, pathway sources, phenotype sources, tissue, also disease databases. So we evaluated based on the same benchmark. And we use the accuracy with the area on the curve of the Roc curve, and also we using like the coverage of data source to measure how rich is a certain data source with respect to the target genes. So here we actually can show that so like the disease genes and also the gene ontology sources, they actually have the high accuracy with the green bar, and also they have high coverage of data sources, which fits our intuition that these are like general sources will work for generate disease benchmarks here. And then we did a case study specifically on the intellectual disability and autism-related genes. So our input here is actually 48 genes overlapping from these two disorders. And we use the data sources as we presented before, which are like work for generally disease, so that we use the geoterms and also the pathway information and the phenotype information and underlying network of string 9. Note that we exclude the disease source here just to avoid that there might be some pre-bias on the disease sources here. And we wanted to do some like novel discovery here, so we eliminate the disease source here. So the final results, we actually get 160 genes ranked on the top with the adjusted p-value less than 0.01. So we did cross-reference this 160 genes with the intellectual disability and also with the autism. And furtherly, we select a few of the related phenotype for these two disorders, showing that we could in total, we could identify 135 genes which are direct or indirectly related to these two disorders. So that will leave us like 25 also genes which are like new to us. It's not like mentioned before. So we see like did literature search see if there's 25 genes are co-mentioned and the literatures. So we find 30 of them are like mentioned in relevant recent studies published after 2011. And then we specifically, the expert in our group are able to identify this specific gene, the AT-CAY genes. So it's functionally related to the two disorders, and it's ranked high in our the top ranked genes. And the function is like related to the cerebellar cortex and the neural diseases, and also neurotransmitters and also axons. So that, so our conclusion is we're reducing this feature and network-based gene superposition method by modeling both type of informations without converting them into each other. And we were able to validate our approach using the disease benchmark, and shows that we could outperform the best performed tool so far. And we're flexible with data sources. So basically, it's like plug and play with the data sources, and we could handle the heterogeneous feature types, like if we have this network-based data or if we have like annotation-based data, we could handle them like simultaneously. And through our final case study, we were able to identify the novel genes actually could be contribute to these two disorders. So it could be used for further testing. And the future work lies in we might want to further identify some patterns on the top ranked genes, and also we want to identify some mechanisms building the network based on the top ranked genes that were identified and on the top of the list. And finally, I want to thank my advisors and also lab members. And I'm ready for questions. Thank you for listening. Thanks.
So the next talk